let us understand history of our recombinant DNA. So here we are going to describe construction of first artificial recombinant DNA molecule. The same was done by our Stanley Cohen and Herbert Boyer. Cohen and Boyer, ये आपको याद रखना है, in the year 1972. So in the year 1972, Cohen and Boyer constructed first artificial recombinant DNA molecule. Now what they did? From bacteria named Salmonella typhimurium. Okay, this is the name of bacteria, Salmonella typhimurium. Here I am drawing. Say for example, this is your bacteria. Of course, bacteria are unicellular in nature, right? And most important thing this bacteria is having is presence of plasmid. What is plasmid? An extra chromosomal DNA other than main DNA. Yes or no? Able of self replication. तो ये Salmonella typhi है. Okay, ये उसका plasmid. And this plasmid, say for example this particular section, this plasmid have, ये जो मैंने यहाँ पे draw करा है, is your antibiotic resistant gene. वो क्या है बेटा जी? एंटीबायोटिक रेजिस्टेंट जीन. ओके, सो कुछ बैक्टीरिया पे एंटीबायोटिक्स की कोई असर ही नहीं होती. क्यों नहीं होती? क्योंकि दैट बैक्टीरिया हैव एंटीबायोटिक रेजिस्टेंट जीन. ओके, वो एंटीबायोटिक को वो लोग रेजिस्ट कर जाते हैं. सो इफ यू आर टॉकिंग अबाउट योर साल्मोनेला टाइफी में ये मैंने ड्रॉ करा प्लाज्मिड, and this plasmid have this antibiotic resistant gene. Okay, so what they did, they particularly Cohen and Boyer cut this antibiotic resistant gene by use of molecular scissors that are none other than our restriction enzyme. उसका एक और नाम मैंने यहाँ पे लिखा है, that is your chemical scalpels. उसको क्या बोला जाता है? chemical scalpels, one and the same. So वापस से एक बार, your recombinant DNA was constructed by Stanley Cohen and Herbert Boyer in 1972. What they did? They cut piece of DNA from a plasmid carrying antibiotic resistant gene in bacterium Salmonella typhimurium. ये मैंने यहाँ पे जो समझाया वो. Cutting a piece of DNA from a plasmid was with the help of course, our restriction enzymes, our molecular scissors. Okay, they are also called as your chemical scalpels. Now what they did, they attached this cut antibiotic resistant gene with vector. Vector in the sense carrier. Vector का मतलब क्या होता है? Carrier. Okay, so once again, the piece of foreign DNA cut from the plasmid, यहाँ से जो आपने कट करा है वो was linked with plasmid DNA acting as vector. Again, as a vector who is present one more plasmid जो आपने निकाला होगा from another bacteria. Okay, so simple as that. ये जो plasmid DNA है act as a carrier. Okay, simple as that. Now what happens? Say for example, this is your vector. Okay, and this is your foreign gene, माने this antibiotic resistant gene. ठीक है, ये दोनों को आपको क्या करना पड़ेगा? ज्वाइन करना पड़ेगा. Yes, this joining occurs by means of an enzyme called as DNA ligase. So what I have written, linking of piece of foreign DNA, मानिए with vector, this thing was done with the help of enzyme DNA ligase. तो क्या करे? Which acts on cut DNA molecule and join their ends. Okay, now this newly formed DNA, yes or no, this is plasmid and it have our antibiotic resistant gene. So this sorry, newly formed DNA having integrated fragment of antibiotic resistant gene is called as recombinant DNA. ठीक है, we are having what, antibiotic resistant gene, ये जो बना इसको आप क्या बोलो, recombinant DNA. Now what we have to do, this recombinant DNA we are going to transfer it into our host cell जहां पे इसका replication करवाना है. Okay, so this recombinant DNA is transferred in E. coli. E. coli over here acts as your host. Its function क्या होगा वहां पे, उसका host, as a host in which our gene carrying antibiotic resistant multiplies. Okay, so what I have written, the vector is used to transfer recombinant DNA to E. coli. To E. coli. Okay, you people have understood malaria, right? What happens in malaria? Your malarial parasite, that is our plasmodium, is carried by mosquito, that is female anopheles. तो वहां पे जो female anopheles है, act as a vector, act as a carrier who carries malarial parasite. Similarly, in biotechnology, vector carries our gene of interest. ठीक है, your vector carries your gene of interest, उसको आपने ट्रांसफॉर्म करा in a host cell, that is your E. coli. Now what happens once it is in E. coli? In E. coli it could replicate in new host in presence of course DNA polymerase enzyme. This you will understand, for replication process there is importance of one enzyme, that is your DNA polymerase. This enzyme is very much essential, right, for replication process to start. You people are going to understand this in chapter number 6. Okay, so अभी के लिए आप इतना याद रखो, DNA polymerase enzyme is utmost for replication to start. Okay, तो host में DNA polymerase enzyme present होती है, जिसकी वजह से there will be formation of multiple copies of recombinant DNA. Okay, so this recombinant DNA multiplies in host cell, जिसकी वजह से you will get multiple copies of your gene of interest. Simple as that. So lastly what I have written, the ability to multiply copies of antibiotic resistant gene in E. coli is termed as, इसको कहा जाता है cloning. Okay, this is called as क्लोनिंग. किसका क्लोनिंग करा हमने, किसकी मल्टीपल कॉपीज जनरेट करी हमने, ऑफ एंटीबायोटिक रेजिस्टेंट जीन, किसके अंदर क्रिएट करी, इन ई कोलाई. सो दिस इज formation of our first artificial recombinant DNA dated back in 1972. Okay, who did this? Our Boyer and Cohen. So this is general description. Let us understand this once again. So here I am drawing your bacteria. This bacteria is your Salmonella typhimurium. To be more specific, right, I am drawing here, this part is your plasmid, that is your circular DNA, and this is the main DNA of your bacteria, that is your Salmonella typhimurium. ठीक है. Now what I am going to do, in this plasmid there is presence of antibiotic resistant gene. So this blue color portion over here represents antibiotic resistant gene, right? Now what I am going to do, I am going to cut. Okay, I am going to isolate this plasmid. I am going to cut this particular DNA sequence. Okay, तो यहाँ पे this is your foreign DNA. Foreign DNA in the sense we are talking about, this is our gene of interest. Gene of interest in the sense we are talking about your antibiotic resistant gene. Simple as that. Now what I am going to do, I am going to take one more plasmid as a vector. Can you see this? Again, ये क्या है? This is your plasmid, and this plasmid over here act as your vector. Vector in the sense it act as your carrier. It act as your carrier. Now what I am going to do? I am going to cut. I am going to cut this gene of interest as well as your plasmid by using same restriction enzyme. By using what I am writing over here, same restriction enzyme. Restriction enzymes are also called as your molecular seizures. Okay, so what happens? There will be fragments over here. Yes or no? Yes, as well as there will be fragments over here. Right? Now what I am going to do, by using an enzyme called as DNA ligase. कौन सी एंजाइम है? DNA ligase, which joins the cut ends. So here I have used your enzyme DNA ligase. What this DNA ligase will do? It's going to join the cut ends of your DNA. Yes or no? अब ये जो प्लाज्मिड है, ये जो वेक्टर है, उसके साथ कौन इंटीग्रेट हो चुका है? Our gene of interest, that is your antibiotic resistant gene. Yes or no? Yes. And this is called as your recombinant DNA. इसको क्या कहा जाता है? Recombinant DNA. Simple as that. Okay. Now what I am going to do? I am going to transfer this recombinant DNA into E. coli. So the process is called as transformation. ये process को क्या कहा जाता है? Transformation. Okay. So here I am drawing your, चलो इससे draw करो. Here I am drawing your E. coli. Okay. This is what transformation means. We are going to transfer our recombinant DNA. The process of transformation we are going to understand in detail bit later, right? तो ये ट्रांसफॉर्मेशन हो गया. ये रिकॉम्बिनेंट डीएनए कहाँ पे आ गया आपका? ई कोलाई में. इन ई कोलाई देयर इज प्रेजेंस ऑफ एन एंजाइम कॉल्ड एज डीएनए पॉलीमरेज. सो व्हाट विल हैपन? क्या होगा? एज सेल divides, what happens? Our gene of interest also replicates. And ये प्रोसेस को क्या कहा जाता है? क्लोनिंग ऑफ एंटीबायोटिक रेजिस्टेंट जीन in E. coli. Similarly, this will also divide to form two more. This will also divide to form two more. मतलब हमने एंटीबायोटिक रेजिस्टेंट जीन की क्या करी? क्लोनिंग. सो ये प्रोसेस को कहा जाता है क्लोनिंग ऑफ एंटीबायोटिक रेजिस्टेंट जीन इन E. coli. Simple as that. Once again, to brush up, what we are going to take? Antibiotic resistant gene from Salmonella typhimurium. One more plasmid who act as a vector. Both we are going to cut by means of same restriction enzyme. इसका importance क्या है? We will understand while studying restriction enzymes. Same restriction enzyme आपको use करनी है, right? Creates cut parts of your DNA. You have to join it with DNA ligase. Join करके जो आपको DNA मिले is called as your most important recombinant DNA. You are going to transform this recombinant DNA into E. coli, where it replicates to form multiple copies, and the process is called as gene cloning. So this is all about your history.